Hello friends, welcome to my channel Botanical Studies. In this video, we will discuss about mineral nutrition. The discussion will go through following points: introduction, macro and micronutrients, criteria of essentiality, and finally mineral nutrition uptake. Introduction. For proper growth and development, every living system requires energy. The energy is supplied by food materials or substances called as nutrition. Depending upon the mode of nutrition, animals are heterotrophic. That is, they derived their food material from other animals or from plants, while the plants are autotrophic. That is, they prepare their own food material. The plants are unique organisms that can absorb nutrient, that is minerals and water through their root system from the soil, as well as carbon dioxide from the atmosphere, and synthesize the food material through photosynthesis. The combination of soil nutrients, water and carbon dioxide along with sunlight allows plants to grow and develop. In plants, growth and development is depends upon the supply of inorganic elements from the soil, that is mineral nutrition. Macro and micronutrients. The complete qualitative analysis of plant ash shows that about 60 different inorganic elements are present in plants. Out of that, 17 are required for proper growth and development in plants. They are known as essential elements. The essential elements can be divided into two groups. First, macronutrients, or it is also called as macro element or major elements. The nutrients which are required by plants in larger amounts, generally present in the plant tissue in concentration of 1 to 10 milligram per gram of dry matter. The 9 macro elements includes carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, sulfur, calcium and magnesium. Since carbon, hydrogen, oxygen are gases and not minerals, while remaining 6 nitrogen, phosphorus, potassium, sulfur, magnesium and calcium are minerals. Second, micronutrients. It is also called as microelements or minor elements or stress elements. These are the nutrients which are required by the plants in very small amounts, that is in traces, equal or less than zero point 0.1 milligram per gram dry matter. The 8 micronutrients includes boron, chlorine, copper, iron, manganese, molybdenum, zinc and nickel. Criteria for essentiality of minerals. Minerals present in the soil enters into the plant through roots. These elements must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plant do not complete their life cycle or set the seeds. The requirement of these elements must be specific and not replaceable by another element. The element must be directly involved in the metabolism of the plant. Mineral nutrition uptake. The mechanism of mineral nutrition uptake in plants is still not known correctly, and therefore various plant physiologists have proposed different theories to explain the process. These theories can be broadly classified into two categories: passive uptake through diffusion, cation exchange and Donian equilibrium; active uptake through role of cell membrane, carrier mediated uptake and phospholipid hypothesis. Before going to these theories, first we will discuss about the plasma membrane. All living cell exhibits the presence of plasma membrane. The plasma membrane is in the form of ultra thin sheets of lipids and protein. The plasma membrane acts as a barrier the cytoplasm from the external environment, and also act as a site of regulating influx and exploits of different substances, including mineral nutrients, between cytoplasm and external environment. The plasma membrane controls the rate of ion movement. It carried out selection of ions for inward and outward movement. Let us discuss about passive uptake. Passive uptake or transport is a movement of ions and other atomic or molecular substances across the cell membrane without need of energy input. Diffusion. Diffusion is the net movement of molecule from an area of higher concentration to an area with lower concentration. There are two types of diffusion processes. First, simple diffusion. The diffusion occurs without the help of any intermediary molecules such as channel proteins or carrier molecules. Second, facilitated diffusion. Facilitated diffusion is the process of spontaneous transport of molecules or ion across the cell membrane via specific transmembrane proteins. Second way of passive uptake is through cation exchange. In the soil, the minerals are present in cation forms. These cations are not static but remain oscillating around the surface of growing root hairs. During oscillations, when these ions comes in contact with surface of root hairs, they are absorbed by the root hair wall or membrane. During such absorption, the cation exchange with hydrogen ion present on the surface of root hair. Due to such cation exchange, the ion of minerals present in the soil are absorbed with greater speed and in greater amount by the normal diffusion process. Third, passive uptake through Donian equilibrium. If a cell containing potassium proteinate immersed in a solution containing potassium chloride, we get a system with K plus and Pr minus ions are on one side and K plus and Cl minus ions on the other side, and the selectively permeable plasma membrane in between these solutions. The plasma membrane is permeable to K plus ions and Cl minus ions, but it is impermeable to Pr minus ions. The Pr minus ions are fixed and not diffusible due to impermeable behavior of plasma membrane towards them. In such system, Cl minus ions will diffuse freely into the cell across the membrane. Similarly, K plus ions also diffuses across the membrane to maintain electrical neutrality. This will continue till equilibrium stage is reached. Equilibrium stage shows the concentration of K plus ions inside the cell is more than the outside solution, indicating absorption of additional K plus ions by the cell. Such additional absorption of K plus ions is required in such a case to neutralize the negative charge of fixed ions in the form of proteinate ions. This equilibrium is called Donian equilibrium. By this process, additional ions, either positive or negative, are absorbed as per the nature of non-diffusible ions. Such absorption is responsible for accumulation and increasing concentration of absorbed ions in the cell. As there is no expenditure of energy, this is the passive uptake of mineral nutrient process. Let us discuss about active uptake. Active mineral absorption is an active process and thus it requires the expenditure of metabolic energy in the form of ATP. Active mineral absorption can occur both along and against the concentration gradient by osmosis or through special carrier proteins in the plasma membrane. When the movement of molecules or ions across the membrane is linked to metabolism, it is called active uptake or active transport. In the active uptake, cell membrane plays an important role. There are three different types of active uptakes: role of cell membrane, carrier mediated uptake, and phospholipid hypothesis. Role of cell membrane in active uptake. Let us discuss about the role of cell membrane in active uptake. For nutrient uptake from the soil to the root system takes place by three different processes via simple diffusion, facilitated diffusion, and active transport. In all these three processes, cell membrane plays important role. Simple diffusion and facilitated diffusion are passive processes, but the active transport process is unidirectional and helps to accumulate ions against concentration. Therefore, the active transport process in the membrane are called pump. The energy essential for active transport is supplied by hydrolysis of ATP. In cell membrane, enzymes required for hydrolysis of ATP are present in the form of ATPase complexes. These ATPase complexes are called proton pumps or H plus ATPase. The plasma membrane ATPase hydrolyzes ATP and use this release energy to pump protons from one side of the membrane to the other side against concentration gradient. In addition to proton pump, the plasma membrane shows presence of pumps for many other ions. For example, pumps for K+, pumps for calcium ions, pumps for chlorine ions, etc. Sodium potassium exchange pump. In addition to unidirectional pump for single element, the cell membrane shows presence of exchange pump. Let us discuss about sodium potassium exchange pump. These pumps are useful for to separate elements against their electrochemical gradient. In sodium potassium exchange pump, the enzyme Na plus K plus dependent is present. In this pump, for every molecule of ATP used for hydrolysis, 3 Na plus ions are pumped out and 2 K plus ions are pumped in. Such pumps are useful to maintain higher concentration of required essential element inside the cell. Carrier mediated uptake. The cell membrane are made up of lipids and protein molecules. Some of the membrane proteins functions as transport protein during uptake of nutrient. These transport proteins are of two types: carrier protein or transporter and channel proteins. The carriers mainly take part in active uptake, while the channel proteins they are take part in the passive uptake. According to carrier mediated uptake theory, during uptake of mineral nutrients, first the nutrient ion binds with the carrier protein, which result in the formation of carrier ion complex molecules. After activation of this carrier ion complex molecule, it moves from one side of the membrane to the other side of the membrane. When the complex reaches to the other side, the ion is released and added into the cytoplasm. The released ions get accumulated in the cell and cannot move out. After release of ion, carrier protein get reverted into its original structure. This is the energy requiring process, so that carrier mediated transport of ion is an active process. Phospholipid hypothesis. The cell membrane is made up of lipids and proteins. The main class of lipid found in the cell membrane is the phospholipid. According to phospholipid theory, in the uptake of mineral nutrients, participation of amphoteric or amphipathic compound is essential. One of the membrane phospholipid is lecithin. The lecithin molecule shows charged polar head region and phosphate group and long tail region with hydrocarbons in the form of two fatty acids. The head region of lecithin molecule is hydrophilic, that is water loving, and the tail region is hydrophobic, that is water fearing. Due to both hydrophilic and hydrophobic regions, lecithin is amphoteric or amphipathic compound. The participation of amphoteric or amphipathic compound in nutrient uptake is important because with such compound both cations and ions can combine. During mineral uptake process, the ion present on the outside of the membrane combined with digital molecule on the membrane surface to form lecithin ion complex molecule. Such complex molecule then moves from outer surface of the membrane towards the inner surface of the cell membrane. On the inner surface of the membrane, hydrolysis of lecithin ion complex molecule takes place due to the action of hydrolytic enzymes on the membrane. Due to hydrolysis, ions are released from the complex molecule on the inner side of the membrane, that is in the cell. Simultaneously, the lecithin molecule in the complex on hydrolysis produces phosphatidic acid and choline. After ions are released, the molecules of phosphatidic acid and choline are utilized again for the synthesis of lecithin. The lecithin molecules moves back to the outer surface of the membrane and there it combines again with mineral ions. By the cycle of lecithin or this phospholipid cycle, ions from the soil are added into the cell. During this cycle, all the reactions required energy, therefore, it is an active process. So, this is all about mineral.